This Week in Virology, the podcast about viruses, the kind that make you sick. From APHL ID Con, this is TWiV, This Week in Virology, a special episode recorded on March 13th. It's not Friday, hopefully, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today here in Atlanta, Georgia, Kathy Spindler.
Hi, everybody. Is this on?
He'll bring it up, right?
Okay.
Thank you.
I want to tell you that here in Atlanta, it's 55 degrees, mostly sunny. That's 55 Fahrenheit. And to get it for you in Celsius, 13. Thank you.
You're welcome.
We prepared. There it is. Yep.
That's the thing on TWiV. You got to know the weather. Also joining me today, so this is a meeting, APHL. I'm going to let others explain what all that means. We have three people who are on the organizing committee for this meeting. On my left, she is from the Orange County Public Health Laboratory, Megan Crumpler. Welcome to TWiV.
Hi, thank you. So excited to be here for our first inaugural APHL ID Lab Con.
And next to Megan from Indiana University Health, Ryan Relich. Welcome.
Thank you very much. And before we get started, I want to congratulate you and Kathy on your upcoming 1,000th episode.
That's right.
In April. So round of applause, everyone.
Can you believe we have almost 1,000? This may be 1,001. You guys want 1,001?
Maybe.
A little enthusiasm.
Or 900.
Oh my gosh. Okay. Also joining us today, she is from the New York State Department of Health, Kirsten St. George. Welcome.
Thank you very much. Absolute pleasure to be here.
And Kirsten is a few hundred miles north of me, right? Is it a few hundred?
Yes, just a couple of hours. Yes.
Yeah, I've been to Albany. Yeah. All right. So we're going to talk about clinical microbiology today. We don't do that very often on TWiV. We mostly talk about virology research, but I think this is a great opportunity to bring what you do to a broader audience. And so I'm going to start by having Megan explain what APHL ID Lab Con is.
Sure. So this meeting, APHL ID Lab Con, as Scott mentioned this morning, gave a nice summary. You know, APHL serves as the association over a variety of governmental labs, local and state public health labs, agricultural labs, environmental, all facets of testing, newborn screening, microbiology, infectious disease, some chemistry, but we are all public health labs. So in 2018, when they were kind of looking at the meeting schedule, they said, you know, we have our annual conference every year in June. We have a newborn screening meeting. So much of our work is dedicated to infectious disease, and we have a lot of infectious disease topics at the annual conference, but we really were in a place where it was like we need a focused meeting on infectious disease, getting a little bit deeper into the science, and how great would it be to combine with our clinical lab partners that we work so well with. Unfortunately, that meeting that was originally scheduled in 2020, which you were going to attend, was canceled. But now that we're in 2023 and we had a great session on laboratory partnerships earlier, our relationships with our clinical labs are even better, and we have great representation here at this meeting and on our planning committee. So this meeting is really designed to dig a little bit deeper into the science of infectious disease and how they relate to public health.
Well, thank you for inviting us. We appreciate it. I think it's a good opportunity. So you plan to have these meetings often?
Every 2 years.
All right. And the three of you are on the organizing committee, correct?
Correct.
It's funny that you're also on TWiV, isn't it?
Indeed.
It's coincidental, I'm sure. I'm absolutely sure about that. So APHL, American Association of Public Health Laboratories.
Correct.
Right? So this is an international organization?
Yes, we do have a strong presence globally in countries around the world. I'm not going to start to list them off, but we have APHL offices located throughout.
All right.
10 total, I'm being told.
You guys enjoying the meeting so far? Yeah. It's good? All right. All right. It was a good kickoff with the keynote this morning, right? That was great. How much more energy could you get?
Right.
All right. So let's talk about the three of you and what you do. But I'd like each of you to tell us all about how you got to where you are today. Tell us about where you're from in your education and training and so forth. And Megan, let's start with you.
Oh, sure. So I am originally from Florida, and I went to University of Florida, go Gators, for my my undergrad. And then I actually was lucky enough to be in an inaugural clinical microbiology laboratory course offered there. Most everyone in my classes was going to med school, but this person said, no med students, only if you're interested in going for higher education, like a PhD. And so I got in that course and I fell in love with my first PCR gel that I saw. And I always have had a passion for viruses. So I was able to go to the Virginia Commonwealth University in Richmond, where I worked on cytomegalovirus vaccine development in an animal model. And I had no idea what I wanted to do when I was done. So I was very fortunate to get a one-year postdoc while my husband finished his pharmacy education. This is when I started listening to the TWiV podcast as I was setting up thousands and thousands of manual ELISAs as a postdoc. And so thank you for getting me through all of those. And I stumbled across a Dee Pettit, who was well known in the public health lab community, and I was able to get my advisor set me up with a 1-hour meeting with her to tell me about her day and kind of what she did. And I couldn't believe the job that she was describing, because it was what I had always written in my head as exactly what I wanted to do, was clinical diagnostics, but for my community. And so I started cold calling lab directors in California, and everything happened very serendipitously. I ended up in Riverside, California, where I was trained. I was able to get the experience to take my board exam, and I have since moved to Orange County Public Health Lab, which is a slightly larger laboratory in California, but I love the public health lab community. I am very involved in APHL and love networking with this group of scientists. So that's how I got here, by chance, like most.
But when you went to Gainesville originally, you were interested in science.
I was. I went with the intention of doing research. I started out as like a chemical engineering major. Wasn't ever going to pass C++ programming, so my advisor said, "How about microbiology?" And I said, "Okay." And
Yeah, people think micro is a soft science, right? It's not, right? No, it's not. How about you, Ryan?
Well, my love for science began at a very, very, very early age. In fact, I don't even remember. But my passion for microbiology really began when I was 10 years old. I recall just seeing microscopic organisms on television shows, and I thought to myself, wow, just look at those things. Aren't those amazing? And my interests grew further, and 2 years later, Richard Preston's book, The Hot Zone, came out, and so that helped me focus a lot of my interest on viruses. And I spent a lot of time in the public library learning as much about viruses and bacteria as I possibly could. And I'm not embarrassed to say that I spent a lot of time throughout grade school and high school behind a microscope in my bedroom rather than out doing the things that, quote, normal teenagers would do. I had gram stain kits, and I would gram stain stuff that I collected from around the house, and I had a decent quality microscope. And so in high school, my junior and senior year, I was actually at a vocational technical school because they had a medical laboratory science program that I was in. And I actually got to teach microbiology to my classmates as a 16-year-old. And so that was really just kind of solidified everything. In college, I did a double major in molecular biology, and it was called medical technology at the time, but now it's called medical laboratory science. And I did a one-year internship and got certified as a general medical laboratory scientist before going off to graduate school. And so my girlfriend at the time, who became my fiance and now my wife, followed me throughout the entire journey from college to graduate school to Indianapolis. And in graduate school, I worked in the lab of Dr. Mitch Balish at Miami University of Ohio in Oxford, Ohio. I actually studied the cell molecular and structural biology of mycoplasmas. I had experience in undergraduate training with bacteria, walled bacteria, viruses, fungi, and parasitic protozoa, but mycoplasmas were this thing that I was told they're just cell culture contaminants and they cause walking pneumonia, but they're really so much more. You can learn a lot about bacterial cell biology by studying mycoplasmas. In addition, Mitch was just, sort of this fantastic magnet for drawing great scientists into his lab. And so I did my PhD work there. And in my last year of my PhD studies, I had applied to a number of specialty fellowship programs in medical and public health microbiology. I was interviewed at 2, and both of them said, sorry, we've accepted someone else. And I thought to myself, well, since I'm in such close proximity, being in Western Ohio to Indianapolis, I'll contact folks at the Indiana University School of Medicine, where a great medical microbiologist, Dr. Steve Allen, and a couple others were. He invited me out for an interview and said, well, we don't actually train PhD folks. We have a MD or an MD training program, but why don't you come out? We can see if maybe we can work something out. And so I was hired there in the fall of 2011 with a stipulation that as part of my employment, part of my fellowship, I had to start a CPEP fellowship program in medical microbiology. And we did that. And in 2013, we got accreditation. Fast forward several years, and I'm now the medical director of the Division of Clinical Microbiology, medical director of a few other clinical labs. I have a basic research lab. I study viruses. I listen to TWiV while pipetting, unless I'm in the BSL-3 facility, in which case I don't. But that's pretty much me.
Okay. How about you, Kirsten?
Wow, 3 very different career paths here. So I was not at all the science kid. In fact, I was a dancer and a musician when I was right through my childhood. I still have those interests really strong. But I became really interested in science actually in high school, especially in biology. And then I became interested in medical science and decided to go into medical technology as my initial degree. And I was particularly interested actually in hematology, until my father arranged for me to have a visit through a hospital lab. And I was fascinated by the hematology machines. And they were showing me the printouts with all the results until the head of the clinical path lab said, yeah, that's really fascinating until you've put a blood specimen through a machine 5,000 times. And I thought, yeah, well, maybe not. And I came out and I ended up working in basic research in research programs for several years. And at the end of several years of basic research, I decided, yeah, this is really not for me. I'd rather do something a little bit more immediate impact. And I left basic research and I went into diagnostic virology in a laboratory. This was in South Australia, by the way. I grew up in South Australia. And a job became available down in Tasmania. And this was in the 1980s. I'm that old. And it was right when they were setting up state reference labs for HIV. And Tasmania is a tiny island state. And they were setting up the state reference lab for HIV at the same time that they were setting up an actual sort of consolidated virology service. And I got the job of actually running that lab on the basis of, not because I had any clinical knowledge, but because I had a tremendous amount of sort of methodology knowledge, because I'd used so many different techniques over the course of the several years in basic research. And it was a tremendously steep, fast learning curve in clinical virology, and I had a terrific mentor, director of the service, Dr. Richard Tucker. They used to call him the walking brain box, and he was. And I was there for several years, and over the course of that time, became increasingly interested in developing new techniques. I loved the diagnostic work, I loved the clinical work, I loved working with clinicians, but I was increasingly wanting to develop new methods and do new things. And I was awarded a scholarship to go to the University of Pittsburgh Medical Center and work on applied research there. And I worked with Dr. Charles Ronaldo, and it was a wonderful collaboration, and he offered me a job. So I went back there the following year and became the assistant director of the clinical lab there. And that ended up being a marvellous collaboration for the next 12 years, during which time I also did a part-time PhD. And by the end of that, I decided I really wanted my own place, my own baby, as we say, my own lab. And so I started looking around, and I was in a quandary between staying in academic medicine or going into public health or going into industry. And the thing that swayed that decision was the opportunity to work at the Wadsworth Centre in public health. And it was a marvellous centre, and just the opportunity to work in a broader context and potentially help more people and work on big projects. And when I saw the scope of some of the projects they worked with, the investigators that they collaborated with and so on, it was too good an opportunity to pass up. So I went in as director of virology, and 4 years later became chief of viral diseases. And we now run a really big comprehensive program with some state national reference labs and a lot of specialty labs and programs as well. It's been a hugely exciting undertaking. They told me when I took the job, life will never be dull. They weren't kidding, and it's been a tremendous last 20 years.
So all three of you are directors of laboratories. Now, we have 2 state public health laboratories, correct?
I'm local in Orange County.
County, New York State, and Ryan, also state or?
Clinical laboratory for a university-based healthcare system.
Okay, so those are 2 of the kinds of public health labs that we can, then there's the commercial public health lab, correct? Like Quest Diagnostics, right?
We would consider those clinical laboratories, the
clinical.
commercial labs, as well as Ryan's lab is kind of an academic clinical. And then Kirsten and I would be considered public health laboratories.
And all of them are represented at this meeting?
Yes.
Correct. Are there any others? Or is that it?
Other types of labs?
Yeah, that would fall under the public health clinical
Yeah,
laboratory?
I'm kind of trying to think about this as a Venn diagram of
So
public
there's
health
there's
which is a subset of which, or
There's like a pyramid that goes along with the laboratory response network that shows kind of CDC at the top, state and local, state public health labs, and then local public health labs. There's also regional laboratories that kind of serve different regions of the U.S. for some specialties like antimicrobial resistance. We have environmental and agriculture laboratories that fall into this public health realm. We're actually really looking at right now about how to expand our public health lab membership within APHL so we can do more of like a One Health approach, thinking about zoonoses and how do veterinary labs play into public health. But the APHL really came from started really with state public health labs. Locals have come in, and now we're recognizing, over the last 10 to 15 years, expanding our membership and including more of the public health focused laboratories that isn't just clinical testing based.
and where would sort of hospital clinical labs fit in? And are some of these labs involved in surveillance and others are involved in diagnostics, and do some do both? Or tell us a little about that.
Yeah, many of us do both. My laboratory, we have a really strong relationship with our clinical laboratories. So I'll give one example. In the state of California, it's required for a salmonella isolate from a patient to come to a public health lab. And so the hospital or clinical lab will take care of the diagnosis of the patient, treat them for salmonella, take care of them. And then when it comes to the public health lab, this starts this whole other investigation and surveillance side. So many of us here are part of the CDC PulseNet program. And so we do whole genome sequencing on our enteric pathogen isolates. Then we compare them to each other, both local, state, and at a national level. We look for sequences that match, and then that may spawn an investigation with our environmental health, who make, we just recently had an outbreak of salmonella at a local restaurant. And so then environmental health goes out, and we had several patients that were identified in the county, not same household. And so they'll go out and look at the restaurant's practices, see if there's anything, in their procedures that would cause some kind of contamination of the food. They'll contact the patients and do contact investigation. They'll look at their occupation. Are they in a sensitive setting? Do they work in a restaurant? Do they need to be excluded from work? And then we're also involved in getting those people back to work if they are excluded. Then we would get the samples to clear them to show that they're negative now for salmonella so that they can go back to work.
Could I just add another comment to that? There's a big range. With public health labs, there's a big diversity in size and complexity, from small public health labs to large public health labs. But the same can also be true for clinical labs. So there are very small hospitals with small clinical labs, right up to major academic medical centres with very large, very diverse clinical labs inside them. So there's this. The old saying, you've seen one public health lab, you've seen one public health lab. The same can be said for clinical labs. There's this tremendous diversity. So while we can, clearly sort of describe the differences in function, there's no one that's an accurate descriptor of all of them. Yeah.
Would some of these clinical ones, maybe in hospitals, have 24/7 staffing?
Oh, yes.
Yes.
Absolutely.
And the kind of work you just described where you're tracking outbreaks. Now, a hospital clinical lab would not do that, right? You would work with the state, presumably?
Yeah, absolutely. So we work hand in glove with state and local health department laboratories, epidemiologists, but the clinical laboratory's primary function is providing diagnostic services for the hospitals, for clinics, for clients that may be affiliated with the healthcare organization. And so our scope of services is a little bit different. We tend to test much higher volumes of samples that come through our laboratories. Like, for example, a patient goes to their doctor with signs and symptoms of a urinary tract infection. A urine specimen will be collected and sent to a clinical laboratory where the culture is performed. And if we isolate a microorganism, say for a multi-drug resistant E. coli, that E. coli isolate, we would perform antimicrobial susceptibility testing, report those results. And if that isolate has certain antimicrobial resistance mechanisms at play, we would then send a subculture of that isolate to the health department laboratory for epidemiological testing. And the same would be true for a variety of other pathogens as well.
So you guys actually have to talk to each other as compared to academic researchers like Kathy and me, right?
Absolutely, and we do on a very regular basis. I feel like constantly hearing from and responding to emails from friends at the public health lab. In fact, we engage them on a variety of levels for various things. We help each other out. Case in point, at the beginning of the SARS-CoV-2 pandemic, I was in near daily contact with our clinical virology laboratory and also the state health department virology laboratory in order to procure samples that we could use for validation testing of our diagnostic assays. We did the same for them. Whenever something comes out, if we get samples, we will provide those samples. That way they can get an assay designed, validated, and up and running.
One of the most interesting things I ever did was the director of the Columbia Clinical Lab, whose name I forgot, but you guys probably know him. He invited me to give me a tour. And I hadn't been in a microbiology lab in 40 years, right? I was blown away by the machines. I mean, there was one where they took urines and it automatically streaked them out, and then there's MALDI-TOF and
Yes.
blood stuff. Oh man, I was blown away. And apparently it evolves continuously, right?
Indeed.
The technology. Maybe we can get into that. But Kirsten, so as a State Department of Health lab, tell us how you operate. What do you get and what do you do with it?
Well, at Wadsworth, it's a very diverse service. So we have several different labs. We have a general lab that receives a lot of respiratory specimens and a lot of exanthemus disease specimens and so on. Our principal mode of detection is molecular testing, especially PCR, but we do have a few other techniques. We also have a specialty encephalitis laboratory, and also a vaccine-preventable diseases lab doing measles, mumps, rubella, that sort of thing, and also a gastroenterics lab, and also a specialty research lab that functions solely as that. So as I mentioned, our principal diagnostic test is molecular testing. And then we, after detection, we do a lot of characterization work on the viruses that we detect, primarily with sequencing, but we also do a fair bit of phenotypic testing. We're one of the few labs in the country that still actually does conventional virus culture in tubes, because we like to actually get those isolates and do a lot of characterization work on the isolate. We also provide those isolates to a lot of collaborators, a lot of basic research investigators, and we also use them ourselves when we're validating new assays, new diagnostic assays, and so on. So it's a diverse area of diverse cross-section of work. And we also run quite a number of applied research programs of our own. And in addition to that, we have 3 national reference centres housed in the labs, one for influenza, one for resistance work, and one for, this is just in virology, and one for vaccine-preventable diseases. So we've also got specimens coming in from anything up to 20 other states, where we're analysing those viruses, isolating, sequencing, and so on, and that data gets fed down to the CDC and so on. So it's a lot of different things going on all at one time.
Why do you get specimens from other states? They don't have the capacity to do what you do?
It's part of the surveillance program for the country, so we're assisting the CDC basically on national surveillance.
So do you get some funding from the state and some from the federal government?
For those surveillance program, it's coming from federal.
Okay.
But you don't get specimens from hospitals, right? Or do you?
We do for other, so we're also funded for other surveillance programs for influenza in particular, and some of those specimens are coming in from hospitals. Yes, we have clinical-based partners that we're receiving from not only from hospitals actually, but from primary care clinics as well. So we have 2 feeds, especially for our influenza surveillance program, with specimens coming from primary care clinics and also from college clinics and student clinics and so on, so that we are getting samples from not just hospitalized patients, which are the more severe end of the spectrum, but also so that we can see what's circulating in the general community in case there's slight differences in those viruses.
So Ryan, when Kirsten was talking about virus culture, you were nodding, because I guess you do that as well.
Yeah, we are among probably the last remaining clinical laboratories, at least in the Midwest, perhaps in the country, that still offer cultivation-based detection of viruses. And so much to what Kirsten said, we do the same type of phenotypic testing on those viruses in order to identify them. And that type of work actually has been instrumental throughout the evolution of our diagnostic services. We're able to take virus-infected cells and use those for generation of controls. We can spike clinical samples with known quantities of viruses. We'll do plaque assay, not in the clinical laboratory, but in my research lab, we'll do plaque assays to quantify them, or endpoint dilution assays to quantify them. And we can inactivate those viruses and then spike them into clinical samples. In addition, I have several collaborators, including you, where we could provide isolates of viruses for subsequent testing.
So tell me the workflow. A specimen comes in, you're in a hospital, comes from a patient with a suspected respiratory tract infection. What do you do with it?
It depends upon the test that's ordered. So our standard of care for respiratory virus testing is actually multiplex molecular diagnostic test. We have an assay that can rapidly identify a number of different pathogens, mostly viruses, but also some bacterial pathogens. And once the results of those are available, we will then report them. That way the clinician can make a rapid determination of how the patient should be managed, whether it's giving the patient antimicrobial agents to fight something like Legionnaire's disease, or withdraw antimicrobial therapy, or even implement antiviral therapy if the patient has influenza or SARS-CoV-2.
So what are the agents in this multiplex panel? Can you name them?
I can, yeah. So we can go alphabetically here. So we'll start with adenovirus, and then the 4 human coronaviruses, 229E, HKU1, OC43, and what am I?
NL63.
Yes, NL63. Can't forget about NL63. In addition to that, enterovirus and rhinovirus. So the assay does not distinguish between those. It targets a region of the 5' untranslated region that does not permit distinction. In addition to that, human metapneumovirus, human parainfluenza viruses 1, 2, 3, and 4, respiratory syncytial virus, influenza A, which it distinguishes H1 from H3 viruses, as well as the pandemic 2009 H1N1 virus. And I said RSV already, I think, yeah. And the bacteria that it detects, influenza B, I can't forget about influenza B. And the bacteria that the assay detects include Mycoplasma pneumoniae, Chlamydia pneumoniae, Bordetella pertussis, and Bordetella parapertussis.
Okay, so no adenoviruses.
Adenovirus was the first.
That was the first one?
Yes.
First one.
I heard it.
You heard it? Okay, very good.
That may have not been in alphabetical order, but
But adenovirus was
it was
first.
close as I could get. Thank you. Thank you.
So, and then are those all PCR based?
Yes.
Or, okay.
Yeah, multiplex PCR.
Okay.
So all at the same time for our listeners, right?
Correct,
It's
yes.
one PCR.
Simultaneous detection and distinction of those agents.
And so, just curiosity, how often do you get more than one positive in an assay?
Frequently. At the beginning of this flu season, if you will, we saw multiple specimens that contained more than one virus. And it's interesting, too. With influenza A, we would see SARS-CoV-2 fairly frequently. But we actually had one patient, a pediatric patient, that had 5 different viruses that we detected. And when you see something like that, you start to question the validity of the test results. And so we did repeat testing, and all 5 of those viruses were detected again on repeat testing. And incidentally, the patient also had an invasive group A streptococcus infection. So our bacteriology laboratory detected Streptococcus pyogenes, and our virology lab detected those 5 viruses.
And so then you said sometimes you would try to culture out some of these viruses to show that are there some that you have a particular interest in, like enterorhinos? Because I know you've supplied us with some of those.
Yeah, I'm very interested in picornaviruses in general, but also...
They're the greatest viruses on the planet.
I agree. I'm very interested in paramyxoviruses also. So anytime we get human parainfluenza viruses, I like to have isolates of those. And even some esoteric for us viruses that we may encounter, like mumps virus. I like the rubella viruses, orthorubella a lot. And there are some viruses we deliberately don't attempt to culture, such as those that require risk group 3, risk group 4 viruses that require BSL-3 and BSL-4 containment.
So, Kirsten, you would do a similar procedure, is that correct?
Very similar, although we don't tend to use that big panel routinely on a lot of incoming specimens. We tend to be, I think, maybe a little bit more selective. We use it on all autopsies. We use it on outbreaks. And we use it on any cases that are presenting with very severe symptoms or an unusual complexity of symptoms. The routine, more routine samples coming in, we would do a much smaller panel, perhaps just SARS-CoV-2, influenza, and RSV.
So Megan, what if you do this as well, I presume, right?
Yeah, there's many similarities.
Okay.
We do also serve, since we're a local public health lab, we do serve our public health clinics, so STD, or STI, HIV, pulmonary disease services for mycobacteria, as well as family health. And then we play a big role in our county correctional facility health testing. So for HIV, STDs, hepatitis, you name it. And then we do influenza surveillance is a big part of the work that we do. And so we are a mixture of diagnostic diagnostic and surveillance services in our lab.
So Ryan, you have a physician who's ordered these respiratory panel. You have a patient with a respiratory illness, and you run the whole panel, and everything is negative. What do you do next?
Well, we would report the results, and sometimes the clinician will call and inquire, well, why was everything negative? And well, I don't know, because perhaps the patient is infected with something that we don't test for. A virus that you're very familiar with, from my understanding, influenza C, circulates still in the population, I have to imagine. And that probably accounts for some of the undiagnosed respiratory illnesses, or there could be even other pathogens that we don't test for that are circulating in the community.
I guess what I'm getting at is, let's say there's a community outbreak of respiratory disease, more than one patient, and everything is negative for your whole panel. When would you do next generation to find out what's there?
Yeah, so we would definitely be in contact with our state public health laboratory in an instance such as that. If we see a cluster of patients who are negative for the traditional pathogens that we test for, that routinely test for, we would reach out to our state health department colleagues and collect specimens and then send those to the laboratory for more comprehensive testing, looking for something that may be flying under the radar, so to speak. And there's a precedent for that sort of thing, by the way. For example, the first case of Middle East respiratory syndrome was actually diagnosed in a patient in northern Indiana, and from Munster, Indiana. That individual was tested for a variety of respiratory pathogens, but because that individual had recent travel history to the Middle East, the laboratorians, as well as the clinical care team in the emergency department and pulmonology services, thought to themselves, well, geez, I bet this patient's probably a good candidate for MERS coronavirus testing. And so the Indiana Department of Health Laboratory then was responsible for making that first diagnosis.
So Kirsten, you were nodding.
Yeah, so there's a number of things that you look at in these situations. The first one, the first thing I was thinking was how well was the specimen collected, which is always a question. But you look at travel history, you look at animal contact, you look at the time period between the onset of symptoms and the collection of the specimen. And then you start to think about unusual pathogens in those contexts, as well as if it gets to the extreme sort of level and the patient is very seriously ill and you've exhausted all other possibilities, you might consider going to metagenomics testing to look for something really unusual.
And that is actually something, too, that many clinical laboratories are starting to offer. They are performing metagenomic NGS in their laboratories. I'm hopeful eventually my laboratory will be joining the ranks of those. But for example, at the University of Washington, Alex Greninger's laboratory, they do NGS testing, and there's various other laboratories that will do the same thing.
So Megan, on a TWiV, we recently did a paper on this using NGS for patients with serious bacterial infections. You think, and they implied that eventually that would be used for everything. You think that's where we're going?
I do, definitely. I think, the technology, affordable technology is not quite there yet. But in cases or in situations where there is unknown pathogens, maybe was it adenovirus that we use? I can't remember, but there are certain labs around the country, there's a couple in California that are known for their ability to do metagenomics on esoteric pathogens and identify them, but it's just not available widely now yet.
Of course, we all have a virome and a microbiome, so you have to distinguish it from that, right?
Yes, and that raises a lot of privacy issues too.
Yeah.
Another big driver too is reimbursement. So for clinical laboratories, we have to get reimbursed for the testing that we're performing, and so it's not to a point where it's affordable for somebody to pay out of pocket for having their entire virome sequenced or microbiome sequenced. And so eventually we're hopeful that insurers will help defer the cost of a lot of that testing. But if we were to perform shotgun metagenomic sequencing in my clinical laboratory today and we were to bill the insurance, we might not get anything back. And so that cost then would translate over into the healthcare consumer being the patient.
Presumably at some point the cost comes way down though, right?
Yes, hopefully, yes.
And then you could do more widespread testing.
Correct.
So, I mean, are you always thinking about the next big thing in terms of a pathogen? Is it in the back of your minds, or you're just distracted by the day-to-day business because all of you are directors? Kirsten? I'm sorry. We'll come to you.
We're always thinking about both. We're always thinking about being prepared for the next thing coming in terms of pathogens, but we're also constantly looking at new technologies and faster and better, more accurate, more sensitive ways to detect them. And also sort of higher throughput, more efficient throughput, streamlining, decreased labor, so sort of improved labor intensity, all of those aspects.
Hence meetings like this, right, Megan? Were you going to say a similar thing?
I was, yeah. Just about preparedness, how it's part of what we are funded through preparedness funding. It's, we're always thinking about if we're buying a platform for a certain test, what else can we put onto that platform? We do have to have diversity because, we typically public health labs, if you may have heard this morning, are the first line for emerging pathogens, things like SARS-CoV-2 or monkeypox. And so CDC releases those assays through the Laboratory Response Network. And then, we hopefully have that extraction platform or PCR platform in our lab so that we can implement that test. Because so that's part of, we have to keep maybe extra equipment or additional equipment, diverse equipment, so that we are prepared for the next test.
Our keynote this morning said, don't squander an emergency build. I guess you all agree with that, right?
Yeah, we're rewriting the pandemic plan right now for New York in light of what we've learned. But we're also always watching things like ProMed feeds and all the data feeds from all the electronic infectious disease reporting and data feeds from around the world, constantly on the lookout for what might come in, especially in major traffic areas like New York, where it's a major port of import.
So I wonder if you could explain that statement a little more. Don't squander an emergency build. What's an emergency build, and was one squandered recently, or what does that mean? I mean, I don't know. So
we never squander an emergency build.
Well, so what's an emergency build? What does that mean?
I think my interpretation is when you have the politicians and the public's attention, and you can more easily justify getting the funding that you need for either continual or ongoing or new infrastructure building. I think would be the easiest way for me to explain it. Yes.
Okay, so don't let this opportunity get away.
Right.
While we have their ears and eyes.
So that we can maintain the system, because there's this history of a surge in funding and then a massive drop, and that drop can go on for a long time, and so you then lose that infrastructure or it deteriorates, and so we need to maintain it, because to rebuild it takes a lot more effort than to maintain it. So if we can just maintain that funding so we can maintain the infrastructure, it's infinitely preferable.
Okay.
So the same speaker this morning said something about politics and public health are inextricable. So public health laboratories are supported by governments, right?
Yes.
And so is that a problem sometimes?
It certainly can be. When politicians kind of make directives to do certain kind of testing or the policies, like for example around monkeypox, you could look at different jurisdictions throughout the country and their response to them was very different based on politics. And so it is certainly something, we try to remain apolitical if we can, because we're here to serve our entire community, but that's not always possible to do.
Right. Is it ever the case that a laboratory says we're going to start looking at this, we're going to test for Mpox, and then a politician will say, no, you're not?
Actually, no, we've not, well, in my experience, we've not had that. In fact, just the opposite. We heard from the Indiana Department of Health commissioner that said what can we do to help you implement Mpox virus testing? And one of the things that happened very early on in the outbreak was, I was vaccinated against Mpox so that I could go into my research laboratory into BSL-3 in order to grow Mpox virus so that I could make reagents to do assay development and validation. And they were very, very supportive of that. And as a consequence, we were able to open one of the first clinical laboratories in the state of Indiana to offer Mpox virus testing.
Yeah, what about you, Megan? Ever had a pushback like that?
Not in my jurisdiction, I mean, unless it's related to funding, but they are pretty open to, we have a very strong STI, HIV program, so we haven't experienced that.
I did hear today at the meeting, though, that sometimes an announcement is made and you guys are surprised. Really? We're going to do 1,000 tests a day?
That has happened.
That has happened.
So why aren't you consulted ahead of time?
That's a great question. I'll defer to Dr. St. George to answer that question.
I'm not sure that I can answer that one either. I think it's maybe that the statements are made and on the hope that their employees can step up.
Okay, that's fine. There is no answer, obviously. Go ahead.
I will say, APHL serves as an advocate for its members, and so major visibility for public health laboratories during the pandemic. I mean, Scott Becker, our CEO, now has an IMDb page because he was in a documentary. So we were many of us in here were interviewed by major news outlets, and so that we're trying to use that exposure to really promote what we do and the work that we do for sustained funding.
Now that you're on TWiV, you're going to get an IMDb page.
All right.
Oh, you think we're small, don't you? Okay. I've some related questions, but first I want to cover something that make sure we don't miss it, and then we'll come back. So you have made your way through the ranks of a clinical laboratory, but for people who want to work in one, you need a lot of workers. What do they need to do? How should they train, Kirsten?
Well, it depends what state you're in. For some states, there are licensing requirements, and you need to look at that fairly carefully, I think. New York has licensing requirements, California has licensing requirements. I'm not quite sure which other states. So depending on what state you're in, you need to look at what the licensing requirements are for working in a clinical lab. And after that, then it's a choice at what level you're wanting to work at. If you want to be a director, you really need to be doctoral qualified, and so you need to look at the path through there. Some people go in through a medical technology degree program. Some people go in through a Bachelor of Science degree program, and so there are various ways you can get there, but you do need to look at the state licensing requirements.
So in terms of education, is there always something needed beyond a bachelor's degree, or
Not
what
always.
would be the
No.
various paths?
Not always. So for clinical laboratories, there's a few different levels. And generally, the first of which is the phlebotomist or medical assistant who's responsible for collecting the specimen, say, for example, a blood specimen. In addition to that, there are associate's degree level types of certifications, one of which is called an MLT, or medical laboratory technician. Generally, those individuals have completed an associate's degree in biological science or related discipline, and then they get internship training where it's kind of a didactic. In the morning, they'll do classroom teaching, in the afternoon, working in a student laboratory. And then the other half of the year is actually spent in the clinical laboratory doing rotations where they can do hands-on work. And after all of that is done, then they will sit for a board of registry examination from whether it be the American Society for Clinical Pathology or American Medical Technologists or another credentialing agency. And so there's also a bachelor's level degree, which is a step up from that. And generally, folks will major in, say, microbiology, biology, or if the institution has a specific medical laboratory science undergraduate degree, like where I went at Clarion University of Pennsylvania, you can pursue that route as well. And following that is another year internship, where you do very similar to the MLT students. And then after that type of training, you sit for a medical laboratory scientist, formerly called medical technologist certification examination. And there are even master's level, for example, a master's in clinical laboratory science. And now there are even programs that offer doctorates in clinical laboratory science. And if you're interested in, if you're a PhD holder, there are certifications that you can get, such as through the American Board of Medical Microbiology. And those are also open to physicians and folks with doctorates in public health and a variety of other doctoral level degrees. Now, for folks that are interested in going to medical school and approaching it that way, they can go through a pathology residency, whether it be a combined anatomic pathology or clinical pathology, or just a laboratory medicine residency. And after that, there's generally one or more fellowships that they can pursue. And for medical microbiology, there are ACGME accredited medical microbiology fellowship programs, which for medical doctors are generally a year long. And those types of folks we train are either infectious disease physicians or board certified pathologists. And after they're done with that training, then they can sit for the American Board of Pathology's medical microbiology subspecialty certification exam or the American Board of Medical Microbiology's certification exam.
And then what would they be doing in a clinical laboratory?
So the doctoral level folks are generally in charge of running the show. So they start off as associate directors or assistant directors, or in some instances can be hired directly as sort of the chief of that particular service line, the medical directors, if you will. Whereas those individuals that have the associates or bachelor's degrees will serve as the technical testing personnel, but they can also serve as laboratory supervisors, laboratory managers, and some institutions also have them as administrative or operational directors as well. So there's a lot of opportunities.
Would you say it's a good career path for
Absolutely,
someone
I
interested
love it.
in science? Yeah.
Yeah.
Yeah, Kirsten, you would agree?
Yes, very much so.
So did each of you have to become directors? Did each of you have to at one point get at the bench there and do virus culture or plaque assay or PCR? Yeah.
Yes, yeah. As part of the training and the experience requirements to sit for the board exam, you do have to have laboratory experience as well as supervisory experience. So yeah, I started as a public health microbiologist trainee in California. So we have an extra certification, as Kirsten mentioned. So that has certain degree requirements, and then there's a 6-month training program, and then you take a state board exam to get your public health microbiology certificate to work in a public health lab. We have a similar track for what's called a clinical lab scientist in California. A little bit different experience and training, but very similar process. So yeah, I definitely started on the bench in my lab.
But when you started, you didn't think you'd be a director one day, or was that your goal?
That was my goal.
It was?
Yeah. Once I
Okay.
met with the director, the assistant director at Virginia, I said, "This is exactly what I want to do." So I did already have my doctoral, so I was coming in,
Got
kind
it.
of at a different place than most, but I was coming in to start that training so that I could eventually sit for the board exam.
You too, Ryan? You wanted to be a director?
Yeah, absolutely. I recall, and now again, here we step back in time quite a distance. I was in 8th grade, and my aunt
I
worked at a
thought
local...
you said you didn't remember anything.
Well, no, it was before I was 10 that I don't remember.
Okay.
But in 8th grade, my aunt took me to work with her. She worked as a phlebotomist in a laboratory of one which was fairly close to where my parents lived. And I met with their clinical microbiology lab director, and I said to myself at that time, I want to do what he does. And if possible, focus on viruses too. And fast forward all these years, and here I am.
Okay. And Kirsten, you also wanted to be a director?
Never occurred to me. Truly, and not from the beginning. And it never occurred to me that I would live in the United States either. Everything was just really just good fortune along the way, and just taking advantage of opportunities when they presented themselves. I have a very non-traditional sort of career path, and I've just been very fortunate along the way to be able to do what I've done. But no, it was much later in my career that I made the decision of, gee, I could actually do that. Wow. And I've been fortunate to have the opportunity to do it. But no, it wasn't something that I was deciding on back in my 20s or actually even in my 30s.
But you didn't mind leaving the bench then, right?
No, I didn't, but I'd been at it a long time by the time I made that decision.
Got it.
I want to ask something kind of different, and that is maybe to have one or more of you describe the most unusual thing that you've done or have had happen in one of your labs. It might be interesting, not only to the TWiV audience, but to this audience, that maybe they can relate.
Well, this is a little unusual for sure, but a couple of years ago at the beginning of the pandemic, I gained a new skill set. So the director of autopsy services at Indiana University Health was looking for somebody to assist with the early COVID autopsies at the time. And I'm not a physician, I'm not a pathologist, and I don't pretend to be. However, I'm well versed with work in BSL-3 with the PPE and everything. And so I volunteered. I went down to the autopsy suite and I assisted with the evisceration of the decedents, and I assisted with various other aspects of the post-mortem process. And so that's a new skill set, was born in June of 2020.
Sure is. Yeah.
And mine is also related to necropsy. So public health labs test for rabies in animals, primarily very small animals and bats. But when at my previous laboratory, we had an alpaca farm that was out in the desert, and this alpaca had recently started exhibiting neurological symptoms, and they suspected that this alpaca could have rabies. And so we usually get, just an animal head, so pretty small, but this alpaca head came in huge, barely fit in our space that we were working in. And so we performed a necropsy. When we did do that, we actually found about a golf ball-sized tumor at the base of this alpaca cerebellum. Did not have rabies, but that was a really interesting day of figuring out logistically how to handle this very large animal head.
So if it's potentially rabid, you have to have certain containment procedures, right?
We do, and so staff are vaccinated against rabies. We do have PPE. We have biosafety practices within our lab to work with the tissues that would contain.
You don't need
rabies.
BSL-3 though, right?
Not for the work that we do. We do because we just have the specimen and the tissue with the virus in it. But we do take special precautions with those. But
all right, Kirsten. Now you have to follow up those.
So there's so many odd cases that come up, and we were talking before last night. It's always on a Friday afternoon, usually preceding a holiday long weekend. A few years ago now, similar, I got a phone call from a colleague, and we had an excavation of a site where they were building a new apartment complex just north of Albany. And the excavation team had discovered some very old human remains. And the owner of the property ordered them to just push them aside and carry on, ignore, which is against the law. So they did that, and they proceeded to the next block of land and found the same, and were ordered to do the same until they came upon just too many human remains to ignore the situation. And they called in some archaeologists who called in some historians who realized that amongst all the remains there were fragments of cloth and buttons and various fragments of material that were able to identify that these were the remains of soldiers from a past war. And when they started piecing together the geography of the site and the identity of the soldiers' buttons and various fragments of clothing, they were able to identify exactly which troop movement it was, and that it was actually the historical previously unidentified grave site of soldiers who died in a well-documented but previously unidentified site from smallpox. And so they had unearthed the grave site of soldiers who died from smallpox. So that, of course, prompted an immediate call to the smallpox team at CDC at 5:30 at night on a Friday. And a massive investigation.
And how did you get involved in that? Because it was near you?
Yes, because it was near me and I was the nearest virologist they had.
Was any smallpox material found?
Not that was still active.
Okay, got it. Well, that's pretty good. It's a good follow-up. Yeah. This is a question just for me, but do any of you do wastewater testing as part of your job?
We did a project on wastewater testing during the COVID pandemic, but the funding for that is stopped. But there are many public health labs that do wastewater surveillance testing now, and that is expanding for other pathogens. So I don't know, maybe Kirsten, do you have any?
We're very involved in wastewater testing. We have a large program in New York State, and I oversee the state sequencing pipeline for SARS-CoV-2 sequence analysis, and we just plugged in the wastewater sequence analysis to that, expanded the testing from SARS-CoV-2 to include several other pathogens in pilot programs for the 22-23 funding year, and we are just in the planning stages for an additional expansion to the program for the 23-24 year. So yes, we've got a lot of wastewater work going on in New York.
I presume polio as well because of the case, right?
Yes, we used it to do the polio investigation and track the movement of that and the circulation, yes.
That's a particular challenge because you need to contain it because we're near eradication perhaps, and there are strict containment measures you need to do, right?
Yes, very much so.
How about you, Ryan?
No, we do not perform any wastewater surveillance testing. However, one of the trainees in our PhD fellowship program, prior to coming to Indiana University, she was at CDC where she worked in SARS-CoV-2 wastewater surveillance.
And there's a lot of programs around the U.S. So in California, we have surveillance for that's run by the state for SARS-CoV-2 and monkeypox. I know that there's in Nevada, I think maybe they're doing Candida auris in wastewater, and so it's really becoming a much bigger, focus and expanding in use. And so, we're all trying to kind of figure out how to grapple with it and where do we go and how do we pay for it.
I guess it need not be an infectious agent. It could be toxins or...
Opiates.
Yes. Okay, so that's quite cool. I want to know how COVID impacted each of you, starting from the first time you said, oh no, and what you had to do. Like the first day you realized this was going to be a real big deal, and what did you do in your laboratories to deal with it?
Well, I was at an APHL conference at CDC when there was musings, and I think it was early March or February. Yeah, and so we were definitely thinking about it. As public health labs, we were, one of the first labs to receive the CDC assay. My kid's school closed on March 13th, and we got our puppy on March 15th. So that was a big change. But really, one of my memories is we got the test kits on a Friday afternoon, of course, and so we came in on Saturday to start running, start our validation. We were hoping to have this validated in 3 days because we were sending all of our specimens to CDC for testing, and it was a logistical nightmare because we had the 6th case in the U.S. in Orange County. And so that, we were just sending so many specimens, and then the testing expanded. and we were just inundated with that. And so we really needed to bring on testing in the laboratory. And you probably are aware of the issues with the kit. And so Sunday morning at 7 a.m. when we came back in, we looked at it, and my molecular supervisor was going, "I know I did something wrong." And I said, "No, you didn't do something wrong. There's something wrong." And I called someone in here who's in the audience, and I said, "Hey, are you seeing this?" "Yes." And so we were on the phone all Sunday trying to figure out what was going on until we identified that there was probably an issue with the assay. So fast forward 3 weeks, we're just sitting around twiddling our thumbs, still sending specimens to CDC, and then then it began when we were able to sort out the issues with the assay. We went from fully manual processes to semi-automated to now fully automated in rapid fire. I think when we were preparing for our CLIA inspection, we had about 22 assays or tests that we had validated for SARS-CoV-2 in about a year, which is just unprecedented for my lab. And so it created, we had to figure out scheduling for staff. We're a Monday through Friday, 8 to 5, and so we had to go through 7 days a week, 16 hours a day with staggered shifts. Specimen management was a big issue. That was actually our bottleneck, was getting specimens into the laboratory, getting the patient demographics and the test order into our laboratory information management system. That was our biggest bottleneck because we had paper test requests coming in. And so a lot of those processes, now we're moving to eTour, electronic test order and result. We've implemented a lot of automation, and so we are ready. Knock on wood.
Was resource scarcity ever an issue?
It was a big issue, and it was what kept us up at night. And I remember a day where we sent out our last and final collection kit out the door, and we didn't know what we were going to supply our epidemiologists with to go collect specimens. And then amazingly, a box came in maybe 20 minutes later, and we started making, I was making kits all the time at my desk while I was on calls. They would just bring me stuff, and we would, right outside my office, and there was definitely an issue.
Did the kits you made work?
They were just swabs and some liquids, so probably.
All right, Ryan, tell us your experience.
Yeah, so it began in very early March, and there were no commercial SARS-CoV-2 test kits available at that point in time, and so we were waiting for something to hit the market rather than develop our own sort of from scratch. But once something became available, we had nothing to validate the assay with. So a good friend and colleague of mine from the National Institutes of Health, Dr. Vincent Munster, emailed me and said, "Hey, we've got the virus in the lab. We can send you some RNA." So they sent it on a Thursday, and it came in on a Friday, of course. And unfortunately, there was a problem with FedEx. So I had to drive after midnight to the FedEx depot at the Indianapolis International Airport to pick up a package of SARS-CoV-2 RNA. And so I drove that back to the laboratory and didn't get back until something after 1 in the morning. After explaining to the folks at FedEx, I really need that box. And they said, "Okay, pal, everybody needs their package." I said, "No, you have no idea how badly I need that package." And so I explained what was going on, and Vincent had sent along primers and probes also to help with assay development purposes. And so we used that material as well as some material we were provided from our friends at the Indiana Department of Health in order to get that first test up and running. And so in mid-March, then we were able to start accepting specimens from Indiana University Health clients and physicians and patients, as well as we helped the Indiana Department of Health laboratories test some. And we also engaged industry in the area. So some friends at Eli Lilly, which is based in Indianapolis, assisted us with production of viral transport media. So that was something that became very scarce very fast, was collection equipment. So while validating and implementing assays in our divisions of clinical microbiology and molecular pathology, we also used our blood bank space. The blood bank within our facility had moved over to one of our hospitals, and so we then outfitted that space with robotic liquid handling instruments, and we began our own in-house viral transport medium production facility. And so we would fill the tubes, we would cap the tubes, put a swab in a bag, and then send that off. And we were making hundreds of those a day and thousands per week. And I have pictures I can show you later of the cases upon cases upon cases of tubes that we would accumulate. And by the end of the week, they were all gone. They were all sent out. So we went through thousands of those per week. And so that gradually then changed. We had the availability of other reagents and other test kits. And so at any given time, we had 2 or 3 live platforms for SARS-CoV-2 testing and another 2 or 3 on the back burner waiting to be activated if necessary. And at the same time, Kirsten and I were on the phone at midnight on Friday and Saturday nights talking to each other about validating new assays and new collection systems and other things. And so she's really been a rock for me throughout the entire pandemic. You know, she was sort of a constant source of, Ryan, everything's going to be okay. We're going to work together to fight back, as well as, friends at other hospital laboratories, clinical laboratories, and colleagues at the National Institutes of Health Rocky Mountain Labs. They were always there. What can we do to help you? How can we support you? Do you need this? Do you need that? And the answer was usually yes. Would you mind FedExing it to us? But now where we are is we have a couple of different fully automated platforms available for SARS-CoV-2 testing, and we perform that testing on a variety of patient types, including symptomatic and asymptomatic, as well as pre-surgical testing and other things that helps our infection control practitioners and our other clinicians manage the care of those patients.
So answer this for our listeners. Why did you do this?
Because I love it. This is an opportunity to help people. I've sort of always been interested in helping people, and this is the way that I could do that. There were a lot of nights where I would text my wife, Amanda, and I would say, honey, I'm probably not going to be home till 2 or 3. And she would text me back, you're doing what you're supposed to do. Ryan, you were made to do this, go do it. And so, I just want to take a moment to thank everybody for supporting me, for helping me, especially my wife, especially my friends, and such as Kirsten and many others, that really sort of refilled the coal chamber so that I could keep pumping, steam out in order to keep going down the tracks.
I think people need to hear that people like yourselves are working for them.
Yeah.
They want them to be safe, right?
Absolutely.
Okay, Kirsten, what's your COVID story?
So I guess starting in January, we were watching, as I mentioned, the electronic information feeds and the data feeds and the news feeds coming in and going, oh, this is not looking good, And then in February, when we had the unfortunate episode with the CDC test, the decision was made to develop an LDT ourselves. And we did that in February. We were very fortunate. We submitted it. We were in close collaboration with the FDA. They were very helpful. We submitted our test on the 28th of February, and it was authorized on the 29th of February, I think, record speed, second test in the country to be authorized. And it was used that night in the early hours of the morning, March 1st, and diagnosed our first case. And that test was authorized to be used by the New York State lab and the New York City lab. And we went straight back in a very few days later and requested permission to distribute it to the molecular virology labs certified by New York State. And we initially specified certain labs that we had very good track records of from our inspections and so on. And the FDA removed the specifications and said, we authorize you to distribute it to whichever labs you consider to be worthy to perform it. So they were very, very accommodating. And we then did that. We distributed the method. There was no charge for the testing, by the way, so we were not being a commercial supplier in that sense, which was the understanding with the FDA. So we distributed the method and immediately to the labs around New York, which helped bring New York up very, very quickly. And then we also went back to the FDA and requested permission to review and authorize LDTs in New York State in place of people having to submit for EUA through FDA. So basically replacing the EUA process through the FDA, through the New York State regulatory system, to hopefully speed up the process and relieve the pressure on the FDA, relieve the workload on the FDA. And they authorized not just New York State to do that, but any state in the country that had a system that could regulate themselves. And so we were able to do that as well, which also speeded up the process. So all that was happening through March. At the same time that we were ramping up testing service, bringing in unimaginable additional pieces of equipment and new tests, and looking at additional ones. I very quickly started receiving a lot of phone calls from a lot of commercial companies asking if I would sign, which I did, collaborative research agreements, non-disclosure agreements, and so on, and have discussions with them about what they were doing to develop tests, because they were starting to want to develop commercial tests. So I became involved in a lot of collaborative work with commercial companies, industry, on the development of assays by industry. And that became a busy time there. And at the same time, I was also, and have always been very interested with the emergence of new pathogens, that material gets stored and information, data gets stored from a very early time on the natural history of disease, of any new pathogen, not just the storage of specimen, but the storage of clinical data wherever possible, so that we don't lose that information about what this new disease is, how it moves through patients, what disease causes it, the pathogenesis of the disease and the course of the disease, and the different manifestations that it takes. And so I was also collaborating with a lot of clinicians in hospitals and so on to that end. So it was a sort of complex combination of work, as well as the regulatory work involved in regulating all of the incoming LDTs that we were overseeing for New York State.
I think even to that end too, though, it's important to note that not just new emerging diseases, but even vaccine preventable diseases that we don't routinely see. For example, measles. Having clinicians aware of the signs and symptoms of measles is very important, and other infectious diseases that perhaps they don't get to see in 2023 on a sort of day-to-day basis. And so, yeah, absolutely to what you said about retaining that clinical knowledge.
Polio is another one.
Correct.
Right? Most clinicians have never seen a case.
Right.
So, but then suddenly you have one.
Yeah.
It's a problem. All right, I have two more questions for you guys. First is there a silver lining? Did you learn something that'll help next time? Because there'll be another pandemic, and is there something that you can use from this experience?
We learned so much, and we've come so far. I really think we've come a long way. Where we're focusing a lot of our funding on, or funding that we've received on, is on informatics and IT infrastructure, because we realized that those were some of our barriers, and so we've implemented inventory management. Because at the beginning of the pandemic, we were always, because of supply shortages, we were constantly counting to see how many patients we could test. So we have an inventory management system. Like I said, we're focused on implementing electronic test order and results for all of our providers, so that we get good patient demographics, quick turnaround of the result back to the submitter. We're even doing things like temperature monitoring in our lab, so we're trying to get away from these manual processes so that we can use our people, because now people have become a scarce resource in clinical labs, as many other professions. We're trying to focus a lot of our resources on workforce development. APHL is focusing a lot. You asked earlier about how do we get into the field, or how do people get into the field? Well, APHL is really expanding their fellowship and internship program. So at all levels, from in college to post-bachelor's to master's to postdoctoral, there's a place for you to get experience in a public health lab at all levels of education, because we are short people, and without the people, we can't do the work, and our people are tired too, after going through all this for the last 3 years, because it just has been, it's been a very stressful environment for many. And so that's where we're focusing our resources right now. That's what we've learned, and lab automation. So they kind of go hand in hand with informatics. We've worked on in our public health lab on anywhere that we can, and this is probably very common practice in clinical labs, but it wasn't for us. With some of our automated instrumentation for results that maybe don't need to be second reviewed, they just automatically go out after they're performed. That was a huge step for us and a big time saver, so we didn't have manual data entry into our laboratory information system. So I think that's where we're really focusing. That's one of the biggest things that we've learned, and we've gotten money to do it. I think we've also been able to show as public health labs that even if you give us a little bit of money, we can do a lot with it. We're very resource oriented, and we can make that turn into some really fantastic projects. And so we hope that continues, that people recognize that we, funding, sustainable funding, we can do a lot with just a little bit more money.
You want me to cut that out of the final thing?
Yes, that would be great. Thank you. I got.
No, because you want them to give you a lot of money. Ryan?
Yeah, a lot of what she said, having multiple sources for different resources. Supply chain issues still plague us to this day. Several years after the emergence and worldwide spread of SARS-CoV-2, we're still experiencing those issues. So diversifying supply chains is humongous. In addition to that, having redundancy in your equipment, your automation, having flexible staff as well, individuals who can be cross-trained from one area into another to help deal with surges of sample volumes and other things. Having very good dialogue with your clinicians, public health partners, administrators, business folks, and those individuals, because they have to be aware too that along with this surge in testing is going to come a surge in spending. And so, letting them all know that there is an issue that's going to arise, that we will have to do things that are sort of outside of the norm of day-to-day business. And so keeping open communication with all the stakeholders in these types of healthcare environments, I think, is absolutely crucial. And even galvanizing existing relationships, starting committees for dealing with virus X, for example, in the future, or whatever the next threat is going to be. So it's really important for the healthcare institutions even to partner with other healthcare institutions, because you need continuity plans for your business. What if you're no longer able to perform testing? Where are those samples going to go? So, establishing relationships with your, quote, competitors, I think, is very important.
Kirsten?
So everything that Megan said, everything that Ryan said, having been through, the 2009 pandemic, Zika, enterovirus D68, et cetera, and now COVID, every single one is different. And I think one of the concerns that some of us have is that too many planning efforts are focused on what happened with COVID and thinking that this is the model, which would be a very erroneous thing to do. It's important to certainly have plans in place and be prepared, but the plans need to be very flexible. There has to be infrastructure in place, and there have to be good lines of communication, but there has to be flexibility, and there have to be people in key positions who have authority to do what they need to do, and clear knowledge of who to communicate with where for what they need. But the plans have to be very flexible, because the next one will absolutely not be the same.
One more. Megan, if you hadn't been a public health laboratory director, what would you have done?
I would have loved to go to culinary school and have been a chef.
Oh, good. All right, Ryan.
I would probably be working for my brother's construction company, running a backhoe or digging in the dirt, because that's something that he and I did as kids, and I could have seen me growing his business alongside him.
Kirsten.
I would have loved to have been a dancer.
A dancer. That might be a new one, right, Kathy?
I don't know. I can't remember.
Did I ever ask you? Actually, save it for number 1,000.
All right.
There you go.
That'd be a
Gives
good
me
question.
time to think about it.
Yeah, right. All right. So that is TWiV. I don't know what the number will be, but it's going to be after 1,000 because I've got them all lined up and we can't mess up. But you can find the show notes for all of these podcasts, microbe.tv/TWiV. And for our listeners, if you have questions or comments, twiv@microbe.tv. Yes, .tv is a web extension. And if you enjoy our work, we would love to have your financial support. We do this for free for everyone, so that knowledge can be free. And so we need your support. You can go to microbe.tv/contribute. The parent company, Microbe TV, is a 501(c) nonprofit. So your donations are U.S. federal tax deductible. Our guest today at APHL ID Lab Con in Atlanta, Georgia, from the Orange County Public Health Lab, Megan Crumpler. Thank you, and thanks for having us.
Yeah, thank you so much for coming and recording here with us today. It's such a great event.
You know, we'll come back another time if you'd like.
Okay,
You have
we'll
another
take you
meeting?
up on that.
From Indiana University Health, Ryan Relich. Thank you.
Thank you very much. And if I may, I look forward to continued research collaborations with you and with Amy as well.
Well, it will be with Amy, because as you know, my laboratory it was closed, but I am remaining as a consultant for Amy. So I look forward to hearing of what the two of you do.
Thank you. Much appreciated.
Kirsten St. George is at the New York State Department of Health. Thank you.
Thank you very much, Vincent. It's been a pleasure, and it's been a pleasure to be up here with friends. Thank you.
And Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks, this is a lot of fun.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank APHL for inviting us here to their meeting. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.